Hello, my name is Dr. Roderick L. Roll. Today, we're going to talk about how to use chi-square analysis. By using the chi-square test, individuals can determine if two genes are linked or unlinked. If the genes are unlinked, we can determine if they assort independently or not. This is an example that we will use to test our chi-square analysis. We have two homozygous individuals that are going to be crossed. They are homozygous for two genes. The first one is big A, big A, big B, big B. That is purple stem, one gene, and short petal, the second gene. We're going to cross that individual flower with another flower that is homozygous recessive for both genes. The genes are green stem and long petal. When you cross those two individuals, you will get a heterozygous individual for both genes. So the F1 is big A little a, big B little b. Then you will perform a test cross. And remember, a test cross will be performed against the heterozygous, for this case, and a homozygous recessive for both traits. After you perform that cross, you will look at the results. If you receive a 25% to 25% to 25% to 25% or a 1 to 1 to 1 to 1 ratio, then the genes are unlinked. This is our decoder key. Whenever you look at the offspring from a test cross, if you have 50% recombinant types and 50% parental types, the genes are unlinked. And remember, unlinked means the genes can be on separate chromosomes, or they can be on the same chromosome, but the distance is so great that they act as if they are unlinked. So the second portion of this decoder key says if the parental types are over 50%, then the genes are linked. That means they are on the same chromosome and they're close enough together to migrate together. So here are the results from the cross. We have 220 purple stem short petals. We have 210 green stem short petal. We have 231 purple stem long petal and 239 green stem long petal. When we look at these, this is the actual data. So now what we have to do is see if the data fits the linked gene narrative or the not linked gene narrative. So you create your hypothesis based on what you observed. So based on what we observed, we're going to create the hypothesis that states two genes are unlinked. Why? Because when we looked at the data, it resembled a unlinked data where 50% of the offspring looked like the parental types. So since the data was close to a 1 to 1 to 1 ratio, or of 50% parental and 50% recombination type, we created this hypothesis that two genes are unlinked. So this is the hypothesis that will be tested using the chi-square analysis. This image shows you that in the original cross we have a parent type that is homozygous dominant for both genes, and we're going to cross that with one that is homozygous recessive for both genes. The offspring that we received are 1 to 1 to 1 to 1 ratio. 50% of them are the parental types, and 50% are recombined types. This chart shows you the same thing. The recombined types are in the middle two columns, and the parental type lookalikes are the two columns on the outskirts of the recombined types. So now you're supposed to compare the observed values with the expected values. This is going to be used to determine if they differ at all. So below the chart, I have the difference of expected versus observed. So the first column, there's -5 of difference. The second column, -15. The third, +6. And the fourth is +14. So there is a little difference. We now need to see if this difference is significant enough to reject our hypothesis. The chi-square equation is used to determine if the data falls within the parameters to support the hypothesis that these genes are not linked. This is called the goodness of fit. This means, does the data fit our narrative? Does the data fit our hypothesis? So if we assume that the hypothesis is true, the hypothesis that the two genes are unlinked, then the chi-square result will determine if the fluctuation that we observed, the -5, the -15, the +6, this is going to help us determine if that fluctuation was minor enough to just be random fluctuation. Or does that mean that our hypothesis is incorrect? So is the data fluctuation random? We determine that by performing the chi-square test. We will now have to use our lecture book, Appendix F. On that page, you will have this chi-square distribution table. We will use this table to determine the goodness of fit. We will locate the appropriate row that coincides with the number of phenotype categories -1. So our phenotype categories observed were 4. So 4 minus 1 is 3. So we will use the row that coincides with degrees of freedom 3. If chi-square falls in the column that is smaller than or equal to 5%, you must reject your hypothesis. So our hypothesis was that the two genes were unlinked. If our value falls between 5% and 0.1% on this chart, we have to reject the hypothesis. Our number was around 2. The number that we're looking at, I highlighted it with the red box. If our number is not 7.82 or higher, then we keep going. Then we don't have to reject our hypothesis. If the chi-square falls in the column that is larger than 5%, then we accept our hypothesis. So in the brackets, anything to the left of 5%, you will accept your hypothesis. Our hypothesis is that the two genes are unlinked. Our number is close to 2.37. The actual chi-square value is 2.13806, which again means it is larger than 5%. So as long as the value is larger than 5%, you will accept your hypothesis. This means that the data fluctuation that we did see is not significant enough for us to reject our hypothesis. So we must accept the hypothesis. The difference between the observed and the expected numbers were small enough to not affect the outcome of the hypothesis. So the data still fits. Now, if the chi-square was in the column that is smaller or equal to 5%, then the variation in the data would have been large enough to accept as random fluctuation. So this means we would have rejected our hypothesis, and we would have to accept the hypothesis that the genes are linked.